Okay, think about the human brain for a second. It's this incredibly intricate thing we carry around every day, the source of everything we think, feel, and do.
Right. And yet, in so many ways, it's still such a profound mystery.
Absolutely. So today we're plunging into a project that's, well, tackling this mystery head on.
Yeah, aiming for nothing less than a really detailed map of the microscopic connections inside just a tiny piece of human brain.
We're diving into the fascinating work of a Harvard-Google collaboration, and our guide, it's an excerpt from The Motorways of Neurons in the Brain, anandthebra.txt.
Exactly. And this isn't just, another study. It feels like a foundational step towards understanding that wiring diagram, the one that powers our consciousness.
Mm-hmm. And we should probably warn you, there are some truly surprising discoveries here.
Oh, definitely. And a real peek into the future of how we study the brain itself.
Okay, so let's set the scene. About 10 years ago, Dr. Jeff Lichtman's lab at Harvard got hold of a human brain tissue sample.
Right. And picture this. It was tiny, like less than a grain of rice, just 1 cubic millimeter.
Wow. Where did it come from?
It came from a patient who needed surgery for epilepsy.
Yeah.
So, necessary surgery.
Okay. But here's where it gets really wild, doesn't it? Inside that tiny speck.
Yeah, get this. 57,000 cells packed in there.
57,000.
And 230 millimeters of blood vessels.
Right.
And the really mind-boggling number, 150 million synapses.
150 million connections in that tiny space. It's hard to even comprehend.
It really is. And apparently, even just looking at it, the complexity was obvious. Dr. Lichtman described it as really beautiful.
That tells you something about the inherent structure, doesn't it?
It does. But that initial awe, well, it quickly turned into a data problem. A massive one.
How massive?
That single cubic millimeter generated 1,400 terabytes of data.
1,400 terabytes.
Well,
what's that equivalent to?
Think about it like this. It's roughly the amount of information in over 1 billion books. 1 billion books. From less than a grain of rice.
Yeah.
Okay, so the challenge then becomes, how on earth do you analyze that?
Exactly. Where do you even start?
So how did they manage to even see inside this microscopic world? What was the tech?
Well, Lichtman's team used some truly cutting-edge imaging techniques. They started by slicing that tiny sample.
Slicing it how?
Into unbelievably thin sections, just 30 nanometers thick, using a diamond-edged knife.
30 nanometers. Can you give us some scale on that?
Sure. A human hair is, about 80,000 nanometers wide? So we're talking incredibly thin, almost molecular level.
Wow, okay. So these ultra-thin slices.
What next?
Then they embedded them in resin and stained them with heavy metals.
Why the heavy metals?
That's for contrast. It makes the structures visible when they use an electron microscope.
Okay, right. Electron microscopy. That's different from a regular light microscope, isn't it?
Totally different. Instead of light, it uses a beam of electrons. And because electrons have a much, much smaller wavelength than light...
You can see much smaller things.
Exactly. You get vastly higher resolution, down to the nanoscale. So they captured thousands upon thousands of these electron microscopy images overlapping them.
Right, like building a mosaic.
Sort of, yeah. But the crucial step was aligning these 2D images in 3 dimensions with incredible precision, nanometer scale precision.
So reconstructing it digitally.
Precisely. Creating this virtual 3D model of that tiny cube of brain tissue.
Right.
And that gave them this unprecedented synaptic resolution view.
Synaptic resolution. Let's unpack that a bit. What does that mean for understanding the brain?
It means you can actually see the individual connections, the synapses, the points where neurons talk to each other.
Communication point.
Yes. Being able to visualize these tiny gaps in such detail is, well, that's what's revolutionary here.
So it's like going from a map of, say, major highways.
To a map of every single street, every alleyway, every pathway in a city. That level of detail.
Okay, so you've got these incredible images, the synaptic resolution map. But like we said, that created this absolute mountain of data.
A data avalanche, really.
How did they even begin to make sense of it? That sounds impossible for humans alone. This is where the Google collaboration comes in.
Exactly. Lichtman teamed up with Viren Jain at Google. He's a senior research scientist there.
And he had experience with this kind of thing.
Yes, he'd worked on mapping the fruit fly brain.
Right.
So he brought that expertise. But even then, think about it, 300 million separate images, trying to identify 150 million synapses.
It's staggering. You'd need something else.
You absolutely needed artificial intelligence and cloud computing. They were essential.
So AI wasn't just like a helpful tool, it was necessary.
completely necessary. They used advanced AI algorithms, these sophisticated programs that learn from data to process it all, to segment it, even colorize the massive data set.
Machine learning doing the heavy lifting.
Definitely. Things like tracing the paths of individual neurons through that dense forest.
Which must be incredibly complex.
Oh, yeah. And detecting all those synaptic connections, mapping the blood vessel network. AI automated huge parts of that.
Yeah.
It really shows how these computational tools are just transforming biology.
Okay, so all this effort, the slicing, the imaging, the AI, what did it actually reveal? Were there surprises in this tiny piece of brain?
Oh, absolutely. Some really striking findings. One of the biggest was about how neurons connect.
What about it?
They found pairs of neurons that formed not just, one or two connections, but sometimes more than 50 synapses between just two cells.
50 between just two neurons. That sounds incredibly dense.
It is. The analogy they used, two houses linked by dozens of phone lines, kind of gets it across. It's a really intense connection.
And what does that mean? Functionally, why so many connections?
Well, that's the big question, isn't it? We don't know for sure yet. The researchers speculate it could be vital for, say, making signal transmission super reliable.
Or maybe integrating different kinds of information, more complex processing.
Potentially, yeah. Or maybe it adds robustness to the circuit. But this kind of super dense connectivity wasn't something people had really appreciated in human tissue before.
Which really highlights why looking directly at human tissue is so important, right? We learn a lot from animal models, flies, mice.
We learn a huge amount, but they aren't perfect replicas.
Right. This direct analysis seems to be revealing architectural details that might be specific to us, like finally getting the real blueprints.
Exactly, not just a translated version. And these blueprints, these incredibly detailed maps, or connectomes as they're called, they have potentially huge implications for medicine.
How so?
Well, the hope is you can compare these connectomes from healthy individuals with those from people who have neurological or psychiatric conditions.
Like autism spectrum disorder or schizophrenia, epilepsy even.
Precisely. The idea is to look for structural differences. Are the connections wired differently? Are there patterns that act like biomarkers for these conditions?
So you could potentially identify these disorders earlier, maybe develop more targeted therapies?
That's the long-term goal. Precision therapies based on the actual structural differences. It's a huge potential step forward.
And it's not just about disease, is it? It could also tell us about how the brain works normally.
Absolutely. Understanding the fundamental architectural principles that support things like learning, memory, behavior itself.
Imagine figuring out the specific wiring patterns needed for memory formation.
Exactly.
Right.
It could open up entirely new ways to understand cognition. What's also really remarkable about this project is their commitment to open science. They made the entire Connectome data set publicly accessible online.
The whole 1,400 terabytes.
The whole thing. Anyone can access it.
That's fantastic. So they're basically saying, here's the data, let's figure this out together.
Pretty much. Democratizing discovery. Inviting neuroscientists everywhere to dive in, explore, test their own hypotheses.
It makes you wonder who's going to make the next big breakthrough using this data. It could be anyone, anywhere.
It's a really powerful approach. And you have to remember, this is just the first step, a tiny piece of cortex.
Right. So what's next? Bigger maps.
Much bigger. The team is already looking towards mapping an entire mouse brain.
An entire mouse brain. How much bigger is that, data-wise?
We're talking maybe 500 to 1,000 times more data. An exabyte-level challenge.
An exabyte. Okay, my head's starting to hurt again.
And the ultimate goal, the moonshot.
A full human brain connectome.
Yes. And that's estimated to be a zettabyte of information.
A zettabyte, which is comparable to what?
Comparable to the entirety of global internet traffic.
Wow. Okay. The storage, the computation needed, it's almost unimaginable right now.
It is. Solving those technological hurdles and the ethical considerations, too, that will usher in a completely new era of connectomics. It'll change how we study the brain.
It's fascinating, too, to see how other scientists are reacting, people outside the core team.
Yeah, the piece mentioned some endorsements. Professor Michael Bienkowski at USC, for example.
What was his take?
He emphasized how crucial this is for moving beyond just extrapolating from animal models. We need to understand the human-specific architecture.
Makes sense. And there was Andreas Tullius at Stanford.
Right. He highlighted the computational questions raised by those really rare but super connected axons we talked about, those multisynaptic links.
Implying they might be doing something computationally very important.
Exactly. And Olaf Sporns at Indiana University, a big name in connectomics, he basically underscored how fundamental this mapping is for understanding memory, thought, behavior, the core of who we are.
So strong validation from the wider field.
Definitely. It really signals the significance of this work.
Okay, so let's try and wrap this up. This Harvard-Google project, it feels like a truly major leap forward in neuroscience.
Absolutely monumental. The most detailed map yet of synaptic wiring in the human cortex, even if it's just a tiny piece for now.
Achieved through this combination of innovative imaging, powerful AI analysis,
And that crucial commitment to open data sharing.
giving us this unprecedented glimpse into our own neural landscape. It's more than just a tech demo.
Oh, much more. It's a fundamental advance. It has the potential to transform how we understand brain function, how we understand brain disease.
It really does. This deep dive into the connectome offers this tantalizing glimpse into a future where we can grasp the brain's organization at its most basic level.
Which leaves us, and you, with a pretty profound question to think about, doesn't it?
Yeah. What comes next? As researchers worldwide start digging into this incredible data set, what unexpected discoveries are waiting?
What secrets about thought, about consciousness, about what makes us human, might be hidden in that wiring diagram, just waiting to be uncovered in the next decade?